without further ado, let me introduce Dr. Jean Kutner. Dr. Kutner is a tenured professor of medicine at the Division of General Internal Medicine and Geriatric Medicine at the University of Colorado School of Medicine. Dr. Kutner received her MD from UCSF in 1991 and completed residency in internal medicine at UCSF in 1994. Subsequently, she completed a NERSA primary care research fellowship and earned an MSPH degree with honors, and completed a fellowship in geriatric medicine at the University of Colorado. She's board certified in internal medicine, geriatric medicine, and hospice and palliative medicine, and cares for patients on the palliative care service and in general internal medicine clinics. And her research focuses on improving symptoms and quality of life for hospice and palliative care patients and their family caregivers. She is co-chair of the NIH-funded Palliative Care Research Cooperative and help lead this amazing Deprescribing trial that we're going to hear about. So thank you so much for being here, Dr. Kutner. I hand it over to you.
Well, thank you, Jen. It's a pleasure to be here with all of you, and thank you to the US Deprescribing Research Network for hosting this. This was actually a kind of actual fun talk to put together. It's been a while since we did this study, and so it felt like a bit like an archaeological dig or anthropologic dig. I'm not quite sure which, because I think I could have probably, before putting together this talk, rattled off, lessons learned, but to actually dig back through and say, well, why did we make that decision? What was going on at the time? It was actually very illustrative to spend the time to put it together. And it also made me realize, I think, how far we've come as a field in the 10 years since we've started the conduct of this trial, because we did start it in 2011. And I think it's very exciting to have the US Deprescribing Research Network to be in existence to carry out this such important work. So I really look forward to the discussion and questions as well. My disclosures are just that I get through University of Colorado. I have research support from NIH and PCORI, and I do serve on the National Advisory Board for the Cambia Sojourn Scholars Program as well. I thought I would start by giving a little bit of context. I'm never quite sure who knows what about the PCRC or the Palliative Care Research Cooperative Group, and then do a very brief overview of the statin discontinuation trial. I'm not planning on diving into the methodology or the results, but just to remind people, I even had to remind myself a bit about it. And then really spend the rest of the time talking about some of these lessons learned. And I've categorized it into 4 buckets: the things we learned from the startup, things we learned from the actual conduct, and then some issues around publication, and then the post the primary analyses, the de-identified data repository and secondary data analysis components of it. In terms of context with the Palliative Care Research Cooperative Group, I want you to think about some of the timing now. So I think even we forget, because like I said, we're now 10 years into the Palliative Care Research Cooperative Group. And what we know is the Palliative Care Research Cooperative Group now did not exist at the time that we were funded to do this trial. So the original funding, the UC4 funding that I list on this slide that we were awarded in end of 2010, early 2011 was both, and I'll talk about that, both to build the Palliative Care Research Cooperative Group as well as to conduct the study. So when we look at, oh, you have all these resources for supporting this type of research, yeah, now we do, but we didn't then. But overall, the purpose of the PCRC is to support exactly this type of research, multi-site, rigorous palliative care and end-of-life research, not just in deprescribing, but in a variety of topics. I put on this slide and on the next one our vision and our mission, and it's really around generating the best evidence and to support that community of investigators who are conducting the research among people with serious illness and their caregivers. And the way that we go about this is outlined here in our mission slide. It's supporting investigators across the spectrum of experience to actually conduct those studies, and then to, as I'll talk about a bit, to leverage standardized data elements and a de-identified data repository to facilitate additional studies, and then to provide those methodologic and other resources needed to help support the research. This is our current structure, and as I said, this is current. It's not 2010 or 2011 structure, and I'll talk a bit very briefly about each of these components. I am fortunate to have some fabulous co-investigators that make up the executive committee with me. Many of you probably know Christine Ritchie, who's also a geriatrics palliative care investigator. She's now at Mass General, and Catherine Pollock, who is at Duke. The three of us form the executive committee for the PCRC. And then the PCRC, as on that org chart slide, is made up of a number of cores and centers. The project coordinating center, which I lead, is here at the University of Colorado, and that includes our operations team that provides more logistical type support. Catherine Pollock co-leads with Tammy Summers, clinical studies and methodology core. Christine Ritchie, in addition to being on the executive committee, leads the investigator development core. And then we have a data informatics and statistics core co-led by Katie Colburn, who's here at University of Colorado, and Salima Megani, who is at Penn. We have a measurement core that is co-led by Antonia Bennett and Laura Hansen at UNC, a caregiver research core that is co-led by Betty Farrell at City of Hope and Nick Dion Odom at UAB. And then our director of our pilot program is Laura Porter who's at Duke. And our director of site engagement, which we'll talk about a bit, is Stacy Fisher, who's here at University of Colorado. So very dispersed, kind of like you guys are, leadership group. Just briefly where we are in terms of members. This was as of March. We actually have more members now, and you can see the growth over time. We have about 560 plus members that are at almost 200 locations scattered across the US, just to kind of give you a sense of where we are now as a network. So back on to and we can, if people have questions about the PCRC, more than happy to answer them, but that wasn't really the focus of this talk. I just wanted to give you sort of the context. So why this slide, as I move to the statin discontinuation or statin deprescribing trial. As I mentioned before, when we got funded back in late 2010, early 2011, to conduct this trial, it was actually the RC4 grant that we got that was part of the Recovery Act funds, the ERA funds. Was actually 3 aims, and it was a 3-year award. So I want you to remember that 3-year award, 3 aims. And this is when you may say, were you crazy? You know, possibly. So conducting the statin deprescribing trial was actually aim 2 of the RC4. Aim 1 was to build the cooperative group. Aim 3 was to evaluate the cooperative group's ability to facilitate the conduct of high quality palliative care research. So the reason for this picture is we joked all along that this was completely building the plane while you're flying it. Or to quote Reid Hoffman, who's the founder of LinkedIn, when talking about startups or entrepreneurs, and many of you may feel this way in the research that you're doing, that it's like jumping off a cliff and building a plane on the way down. So just to give context of in which we were conducting this trial, and like it's a completely separate talk about building a research cooperative group from scratch. So we're going to focus on the trial itself. So the research question for the trial was really a safety question. Is it safe to discontinue statins for patients who have less than 1 year to live? Somewhat interestingly, this research question came about. We had held a planning meeting in February of 2010 around this whole idea of building a palliative care focused clinical trials cooperative group. And as part of that original planning session back in February of 2010, we said, well, let's draft out a straw man trial. What if we were going to get funded to what would we do as a first trial? And this actually came out of that initial brainstorming where we said, what are important questions in palliative care that you really need a multi-site study to conduct? And this was the research question that came out of that. It was a multi-center, unblinded, pragmatic trial with randomization of eligible population. Simply, they were either randomized, they had to have been on statins, and they were either randomized to discontinue or continue their statin. And the outcomes that we were interested in were survival. Remember, because we were looking at it in terms of safety, survival, cardiovascular events, and then we did also were interested in the impact on the patients themselves, so quality of life and symptoms, also number of other medications taken, and health resource use, utilization and cost. Our population were all adults. They had to have been on a statin for at least 3 months, and they had to have an advanced life-limiting disease, which we defined both using the surprise question that the treating physician would not be surprised if the patient died within a year. They did have to have a best guess life expectancy of more than 1 month. And then we defined advanced as they had a decline in, this is their performance status to less than 80% in the last 3 months. And we did allow people in the study who had a legally authorized proxy to also enroll. They didn't have to be cognitively intact. We did exclude some populations, basically those populations that had a contraindication to either discontinuing, i.e. the first bullet, or continuing a statin, or if the patient or proxy were not willing to consent, or if the treating physician was unwilling to allow enrollment, which we may want to talk about in terms of lessons learned about gatekeeping and exclusion criteria. So just in terms of to get to the numbers, we screened about 1,600, almost about 1,000 of those were ineligible. So we were down to about 600 or so, 660 that were eligible. There were a number of barriers. If you look over here, we'll talk about lessons learned about recruitment barriers of either the physician or the potential participant being unwilling to participate, which ended up with our final enrolled number of 381. Keep that number in mind because there was a lesson learned there too. We ended up with 15 sites kind of scattered across the US. As you see here, Colorado is the rectangular state there in the middle, in case you're wondering. But it was primarily academic medical centers, although not all. For example, Duke, Colorado, UNC, UAB, Beth Israel, Wisconsin, Mount Sinai, WashU. We also had some community-based sites. There was a Kaiser-based site, several hospice and palliative care based sites kind of scattered across the US. Here was our analysis plan. It was an intention to treat approach, and we were again looking at safety analyses, initially looking at time to event. And our median follow-up, as you see, was about 18 weeks. So brief, quick overview, and then brief, quick overview of the results. Oh, that's hard to see at the bottom, but it was published in JAMA Internal Medicine in 2015. We did enroll 381 patients. You can see the patient characteristics there. And this was very top line results that it was basically bottom line was safe to discontinue statins, no difference in rate of death within 60 days. All right, there is your very quick overview of the study. And now we will dive in probably the more interesting part, which is what did we learn? Lots. If you to think about the study startup lessons learned, and this first one feels like something that we learn and then relearn and then relearn again as investigators. And I honestly had actually forgotten this as I was digging back through the archives of my files to see what did we actually learn here. Like I said, I completely forgot this, but it happens to us all the time. Our initial proposed budget was cut by 25%. And so what did we do? We cut our budget. So we allocated it out across the sites. We changed from in-person to phone-based follow-up. But we didn't actually cut our scope of work. And I think this would be sort of an interesting point for discussion that's not unique clearly to deprescribing trials, but I think we do this all the time as investigators. We're so committed to the research question and the study that we've proposed that we somehow magically think that we're going to still do the same scope of work that we said would take X budget. And then when our budget gets cut by, X minus some percent, we still say we're going to do the same scope of work. And I think this was a really important lesson for all of us in terms of what can we really do with the available budget and making those, I think, really hard decisions up front, and then, negotiating and navigating that with the funder. I think what we should have said is for a 25% less budget, here's what we can actually accomplish in terms of scope of work. So there's lesson number one. Lesson number two was around IRB. Remember this was pre single SRB, single IRB days. We learned a lot of important lessons around navigating IRBs, particularly around studies in the palliative care and end-of-life population. We ended up starting with the University of Colorado IRB, since that's where I was, or I am, as well from the Duke IRB, since that's where the primary grant was, since Amy Abernethy was at Duke at the time. And we got a lot of pushback early on from the IRB about this being a vulnerable population and the potential harms of this type of study. And the important lessons learned there were actually around having dialogue with your IRB, developing a strong relationship with the head of your IRB, and to be able to negotiate some of these things up front and really address both this type of study design as well as work in this population. And so the other lesson learned is we turned a lot of these lessons into scholarly products. So this was a paper that came out of that work on working very closely with our IRB, and even up front working with the IRB even at the time of study submission, so that we're not getting tripped up along the way. Lots and lots of lessons around a site and site PI selection, and I'm going to show you in a bit the differential recruitment across sites with names of sites made confidential to protect the innocent or something. What we learned a lot in the site and site PI selection, we've carried forward actually into a number of the processes that the PCRC now uses when it works with investigators to identify sites for their studies. And these actually are some of the questions that we now ask. When we were starting this up back in 2011, we were kind of, and any willing comer who said, yes, I'm interested, and yes, I want to serve as a site PI. for this study. Some important questions to ask as you as investigators are finding other sites to participate in your studies, or if you're being asked to serve as a site PI for a study, is to really understand the type of care settings, what will be used as recruitment sites. Am I recruiting from an inpatient hospital service, from an ambulatory care, from a nursing home, from a home-based hospice or palliative care service, and what are the unique needs of each of those settings in terms of recruitment, and knowing within those settings also who are going to be both the gatekeepers and champions. I mean, I've experienced this over many years of doing community-based research, even prior to PCRC being in existence, is that the person who agrees to do the study or to serve as a site PI may or may not be the person who really is on the front lines, that is going to be actually making those decisions around whether patients will really be getting to your study, and understanding that local ecology is so important. It taps in with the second large bullet around the site PI. One of the things that we found kind of consistently both in this study and then moving forward with the PCRC is that we may have a palliative care investigator who is very enthusiastic about and wants to be the site PI for the study, but they are not actually the one who really has kind of the control over that patient population. So it does finding out whether that site PI really has sort of the ends, the relationships, the power, or whatever's needed to get to whatever that patient population is that you're actually interested in. I think systems are getting better to be able to identify potential study participants, such as using the electronic medical record, but really understanding what is possible at each of your sites as well, in terms of identifying the patient populations and identifying ahead of time what those hurdles might be. And I've listed out some here on the slide. And then once you have good sites, good site characteristics, also helping your sites in identifying and hiring the right CRCs, being clinical research coordinators. I think particularly in the palliative care and end-of-life population, it is a very special skill set of people that are comfortable with and have the skill set to recruit patients to studies. It takes a lot of empathy, excellent communication skills, and also one of the lessons learned along the way is, especially if you have inexperienced and younger research coordinators who may be some of your best research coordinators, they haven't had a lot of contact with seriously ill patient people or death and dying. So there can be a lot of support that they need. They can be very effective, but just because we are comfortable, and especially those of us that are clinical with this population, does not mean that your CRC will be. And we've definitely learned that lesson the hard way. And then the need to train and retrain again, and building that into your budget and your timelines, because over the course of the study, you are going to lose study staff, you are going to lose sites, you are going to add sites, and we don't tend to think about planning in both the timelines and the budgets for all that training and retraining. So the positive outcome from all of this is we have used these lessons to refine our site PI for a PCRC perspective and site selection process, as I mentioned. And that's something that, as I mentioned when I was showing the pictures, that Stacy Fisher oversees in her role. We also created, oops, there we go. I have created a number of resources, which I put in the links for, which I'll talk about in a minute. Thinking about outcome and measure selection. This is, again, not unique to deprescribing trials, But one of the things that we often get asked about this study, and I'd love to hear your all thoughts as you're thinking about deprescribing trials, is we specifically decided not to include any lab or physiologic measures. And this was really for logistical reasons. If you think about our study population, most of whom, or many of whom, were home-based, and also, we're not regularly getting labs. In fact, having blood drawn may not be consistent with their goals of care. That's why we made that decision. We have, as we were writing up the paper and had discussions afterward, people said, well, why didn't you follow, lipid levels or other labs? And I know some of you on this call, I've had discussions around deprescribing trials, say of diabetes medications or blood pressure medications, where having lab or physiologic measures may be more important. But having that being a purposeful decision and understanding why you're making that decision, I think is really important. And then especially in this population, there's always the would be nice to measure versus what it is you actually need to measure. There are a few resources available for you, like I said, through the PCRC along these lines. Something that Laura Hansen and Antonia Bennett have put together in the measurement core section of the PCRC website. There is now a measurement library, so you can go looking there rather than starting your own search. And we also have our standardized data elements for those sort of core measures that are measured across multiple studies. So this is just a screenshot from the measurement core, and you can see what's available there, the measurement library. There's some systematic reviews of instruments, some work around using patient-reported outcomes. And then this is just a screenshot of our standardized data element form, just to give you a sense. It's longer than that, but just as, if you click through, if you go to the website, you'll find that. All right, study conduct, lessons learned around recruitment, site performance, working with your DSMB, and then adverse event definitions and adjudication. I think probably recruitment is probably sort of the bane of all of our existence when we're doing studies. It's like James Murphy's rule of study recruitment, that all those patients who meet your eligibility criteria, that when you look at your own population or look at your study sites and ask them to make an estimate of how many patients will be eligible that meet the study criteria, I swear the minute you start study enrollment, all those patients disappear. I don't know what happens to them, but they disappear. So we'll talk about some lessons learned in recruitment. And what we turn this into, this is a screenshot from the PCRC website, is that under the resources section, there's a number of training videos and documents that get at some of these issues. Some key lessons learned: language matters in both describing our studies as well as in recruitment. Think about the language that we use all the time versus what particularly patient populations may think about. So we talk about serious illness, end of life, at least in our population, all the time, and don't really think twice about it. But you think about the patient population that maybe nobody's actually ever told them yet that they have a limited life expectancy. We had a number of situations where, as our research coordinators were consenting or talking to patients about the study and consenting for the study, patients were like, what? Nobody told me that I was, seriously ill or near the end of life. The other thing that we found, and this has changed my approach with my primary care physician hat on, is patients would say, I can't discontinue my statin. My PCP told me, or my cardiologist told me that I have to take it for the rest of my life. And you think about what that language then implies. If somebody says, no, it's actually okay for you to stop it, what does that mean in terms of somebody being near the end of their life? So I think I was probably guilty of using that. Yeah, you're going to take this for the rest of your life language prior to being a PI on this study. It also, language matters in terms of the scripts that we use for our research coordinators and the scripts that we use as we're talking with the primary treating physician. And some of that we have, like I said, put into these resources. There are training videos on the website that came out of this experience that talk about, this one is the informed consent process and discussion. There's also a training video on participant recruitment. There's one on the CRC, the research coordinator approaching the primary treating physician, and then one for PI, site PIs approaching the primary treating physician. And then, as I mentioned earlier, recruitment is an art. Some of the things that we have put on the website include some handbook that is for clinical research coordinators. You see a link here, and I think I have, right, here's what that looks like. Marlene McKenzie was actually our lead clinical research coordinator for the statin trial, and Rachel Kendrick is our operations lead, and they put together this very nice handbook that addresses a number of these key issues, particularly around supporting our clinical research coordinators in that very difficult role of being out on the front lines, of really being the face of our studies. And I think really can't emphasize that enough, that we really need to fully support our research coordinators, set them up for success, and regularly reevaluate our recruitment processes. And then in the vein of the other lesson of turn things into scholarly product, this was a paper that came out from the PCRC back in 2014 that describes some of these experiences and lessons learned. Okay, site performance. How many of you have been in the situation where you have sites that differentially recruit to your study? A couple things I want to talk about here. One is how we pay our sites, and two is how do we and when do we decide to pull sites out of the study, and when do we decide to add sites to the study. And I think this comes up with just about every multi-site study I've ever seen. So this is a table. This is, we would follow our monthly, that you see in the top right, our monthly report of study enrollment. You can see here that we started enrolling, you see at the very bottom, we started enrolling in June of 2011. That was at one of our prime sites. And this table was pulled from about 13 months later, so from July of 2012. And at this point, we had 11 sites for our study. You can see on the left there. I'm going to point out to you in what's that, 1, 2, 3, fourth column down, that our target enrollment at that time was 1,200. File that away in the back of your head for a few minutes, because you're going to say, well, wait a minute, you just told me you enrolled 381. So that's another lesson learned. What I want you to pay attention to here is that we started out with each site setting different targets. That's in what the third column over, that we had different targets, third and fourth. We had different targets for each of our sites, depending on their size, when they started enrolling, because you can see from the second column that sites started enrolling at different times. You can see there it was differential enrollment, not surprising. They started enrolling at different times, but what I actually want to have you pay attention to is the enrollment rate. And this is the number of patients that were being enrolled in the trial on a monthly basis. And you can see there is significant variation there. So this table and the monthly reports leading up to it was actually what led us at about 13 months into the trial to change the way we were paying sites. And it's actually the way that we now recommend, we being the PCRC, that investigators pay their study sites. We started out in the way that I think maybe all of us were, except those that grew up doing drug trials, were taught as you budget an X budget per study site, and that's for the investigator and their research coordinator, et cetera, et cetera. So we had a fixed payment model up until 13 months into the study. And what we found is that for, say, this site number 6, that was one darn expensive enrollment. That enrollment was probably a $50,000 enrollment in terms of what we were paying that study site on a fixed model, as opposed to site 1. And granted, it's in different study that they'd been enrolling for different study times. But the site 1, it was more like a, $1,000, $1,500 per patient enrolled. So what we changed to after watching this enrollment, recognizing that we actually could not continue to afford to pay site 6, let's just say, I'm going to make up some number, $50,000 a year for their one patient that they had enrolled, and we needed that budget to further support, say, sites 1, 2, and 3 that were enrolling at a higher rate. Is we went to a change in the way that we funded sites, which was new sites would get a $28,000 startup payment that would pay for the time for getting IRB or whatever else needed to be done to get started, and then went to more like what you see in drug trials. We were paying then $1,500 per enrolled participant. And it allowed us to reallocate the funds in such a way that the higher enrolling sites were actually getting more money. It was controversial at the time, and I'd love to hear people's thoughts on how you all are paying your sites, but it is the way that we now move forward with the PCRC. This is the enrollment table then about 1 month before we ended enrollment. It has all the original sites, the original 11 plus the 4 sites that we added. And so with this, you see that by adding those 4 sites, we were able to get an additional 36 enrolled participants. Note the number down here of 360. I'm going to come back to that. Our target did change. But what this shows is that what happened when we basically stopped supporting some sites that were just not performing. You can see the same low performing site here did a total of 4 patients for the entire trial is what they enrolled. So thinking about how you're going to pay and incent your sites for recruitment, I think, is another really important lesson. All right, now I'm going to get to the point where why did that first table have 1,200 and our second table had 360? A couple of lessons learned here, which has to do with really understanding the characteristics of your patient population. And really be thoughtful about how you populate your DSMB. We were very fortunate that we had a very engaged DSMB that worked with us closely on this. So here's what happened. So when we first designed the study, we had estimated that survival of our study population, median survival would be about 3 months. And that is what we calculated our sample size on, which got us to a sample size of 1,200. And remember, our original endpoint was actually median survival. Note that is not the result that I showed you earlier. The result that I showed you earlier was proportion who died within 60 days. So I'm about to get to how that happened. So we were watching our study population, and we were realizing that our study population was actually living longer than we had originally thought. At about 15 months into our study start, we began discussions with our DSMB, because what we realized is that the median survival in our study population, rather than 3 months, was 9 months, which means that to get to our original calculations of using median survival as our primary outcome, we would need a study sample of more than 30,000. Imagine Amy and my response when we got that from our statistician. Talk about not being achievable, even if we'd had our original full budget. So we began a discussion with our DSMB about 15 months into the study, showing them these data and reviewing what our options were at this point. We worked closely with our DSMB and our study statisticians and developed multiple scenarios of in terms of different endpoints to use that were still true to the original study design and would answer the original study question, and what that impact would be on sample size. We presented multiple scenarios to the DSMB and ended up on revising the primary endpoint rather than median survival to death within 60 days of study enrollment. And this was informed by the scenario planning, as well as looking at the existing literature on when there might be an expected outcome in this population. Redoing that sample size calculation is what got us to the 360. This was not just communicated with NINR, who was the funder at month 17. Because this was part of those DSMB discussions, we were keeping our program officers up to date on this all along. We did end up with getting full approval, and then I think some key pieces here were, like I said, working closely with the study statistician, closely with the DSMB, and also document. And this was probably actually the easiest bit of my archaeology when I was putting together this talk, because we had such good documentation from both our study statisticians and our DSMB of each of the steps along the way. This is not something that you want to try and reconstruct, because then also imagine when you're looking forward to actually writing the paper of having to explain this. Okay, adverse event reporting. This is probably a shorter section. Some kind of key lessons learned on that is we tend to think about any bad outcome as an adverse event, and making sure that you're really clear in your study design about what are predefined study outcomes. And I've listed ours here on the slide versus what is an adverse event. Really understanding the definitions there. And in our case, the definitions actually changed. The AE reporting categories as defined by the FDA changed about 3 months after we initiated study enrollment. So we actually updated all of our forms to reflect that. Thinking about your study population and what data will actually be available. We found in our palliative care and especially our hospice study population, a lot of detailed information about what might be adverse events was not well documented. And also having an adjudication committee that can really help with this, and particularly to address that question of whether an adverse event is related to trial intervention. I would say this is a place that's ripe for further exploration and writing, particularly in the palliative care population, in terms of what actually constitutes an adverse event in the palliative care population or the hospice population, and how those should be both defined and adjudicated. So if there's anybody in this network that is interested in that, it is an area that is ripe for some further definition, because we'd be much better off if we were all using some of the same definitions. We felt that we were in very uncharted territory back in 2011 to 2014 when we were conducting this trial. All right, I'll wrap up with my part with the kind of last 2 sections. One is around publications, and especially when you're conducting multi-site trials with multiple authors, it is so much better upfront to have a clear agreement around how authorship will be defined. This is the PCRC has on our website. I put the link there, our authorship protocol. You see that we're now on version 2.0, which is technically the third version, because we've of course learned lessons along the way, but having that written out standard and ahead of time, I think is incredibly helpful and useful, so that you don't have those arguments about when somebody can be an author and what it counts as to be an author. One very practical thing that we learned that I think is probably less of an issue now with more use of like Google Docs and other shared editing is, I think Amy Abernethy and I still have nightmares about when we got back the edits from all the authors for the main paper, when and we have multiple authors, and it was in tracked changes, but multiple versions of track changes. And for any of you that have tried, I think I literally sat on my living room floor with like color copy versions of each. This is going to sound like, like so 2013 to try and rectify all the edits. So having clear version control, and I would use a shared editing platform. Don't do what we did. And then there's always the primary paper, and then early on mapping out what are going to be those secondary papers and who's going to be accountable for those, so you can have parallel work going on to get those publications out. All right, the last section will be then about multiplying the use of your data. As I mentioned somewhere earlier on here, that the PCRC has a de-identified data repository, and the statin trial data are in the de-identified data repository. The advantage here is that then your data can have a multiplier effect, can be used by others to conduct secondary analyses, or to be used as pilot data for other studies. What we have learned through the process, though, is most of us don't think from the very beginning when we're setting up our study databases or planning for sort of the study closeout that we are going to be putting our data into a de-identified data repository. We have worked closely with a number of investigators to help them prepare their data to put into the de-identified data repository. So that is something that I would encourage people to be thinking about from their beginning study planning, especially as they're working with the statistician and data analysts, as setting up the databases in a way that they can be easily cleaned, de-identified, to be able to be put in a de-identified data repository. And then the other lesson learned the hard way is that when somebody else is analyzing your data, they may discover that you made a mistake. And this is exactly what happened to us. This was now several years ago, where we had an investigator using the de-identified data from the statin trial, was conducting analyses, was actually responding to a reviewer's comments. So a reviewer had asked about how missing data had been accounted for on one of the outcomes. It was one of the patient-reported outcomes, quality of life or something. And it was amazing that we had a master's student who was working with us on our de-identified data repository. And so she went to answer this investigator's question. And in the process, she discovered that we actually had a coding error in our calculations of some of the secondary outcomes from our original trial. I won't go into the details of what the coding error was, but we dove into it, and there truly was a coding error in how we had calculated for missing data in 2 of the patient-reported outcomes that were our secondary analyses. So we learned a lot of important lessons from that. One, what do you do? So we now had the primary paper published, as well as I think two or three other secondary analyses. And so, we worked with our statistician, redid our primary analyses, and then redid the analyses for everybody that had done a secondary data analysis. Fortunately, we discovered that while it changed some of the absolute scores, it didn't change any of the directionality or any of the conclusions, but we did learn what is the process then when one discovers such a thing. And this is the erratum that was published. Remember, our original paper was published in 2015, so this was what we published in January of 2019, describing the coding error and making the changes to the original published paper. So the things that we've done now with the de-identified data repository for the PCRC is we are getting the coding and make sure we have that captured, as well as have a process for reviewing it. And then the other lesson from our team is those of you that work in quality and safety, we took a just culture approach to this and said, we were all in on this. We all take responsibility for it, and we will all deal with it together. We had no finger pointing, and all the primary authors, or all the authors of the papers, and the investigators that we worked with around their secondary analyses were terrific around that. So I'm going to end there, and this is a summary kind of what we went over, but I would say, and especially in reflecting on it and putting together this talk, we learned a lot of lessons in doing this trial. We were a bit in the Wild West doing this. It's the first time a deprescribing trial had been done in palliative care, and a lot of it now forms, how we function as a PCRC. So I look forward to hearing your thoughts. I've seen some things with my side of my eye there coming across in the chat, and would love to hear your thoughts, or answer questions, or learn what you all have figured that it's probably much better than what we did way back when.
Thanks, Dr. Kutner. Thanks, Jean. Really incredible work. As you mentioned, still hugely impactful study, and of course the PCRC as well. So I'm going to just go through some of the comments and questions. The first one that I'm going to mention is going back to the budgeting and the adjustment, and whether you could say how receptive the NIH is to reducing scope of work when they reduce your budget. And there was some chat about that as well, but if you had experience with that.
It is a difficult navigation. I guess so. The thing I would say is that this is where I think having a good relationship with your program officer is probably probably the most important thing, because I think, as we all know, we have different relationships with different program officers. I would say it is difficult, and that doesn't mean we shouldn't do it. We need to be honest about what we can and can't do with our available budget, though. And I think then the other thing to be really careful of is to then, if something gets changed, just make sure you get a new notice of grant award too. Because our NOGAs are really, lines out what it is that we said we were going to do. And if your NOGA is written with your existing, your prior scope of work and budget, and then you navigate or negotiate doing something different, get a new NOGA too.
Well, great advice on that. There's some comments that, in addition to the resources that you provide, there are other resources. For example, the U.S. Deprescribing Research Network has some resources for working with IRBs and templates, as well as data and safety monitoring boards, and that link is in the chat as well. And there is work, I see here that some folks are associated with the US DEN are doing some work on how to adjudicate adverse events in deprescribing
Thank
trials.
you.
Cynthia Boyd and Liz Bayless. Also, our Pepper Center at Mount Sinai has done some work on adverse events in studies of patients with serious illness and how the usual kind of FDA criteria don't really make that much sense.
Wonderful. Thank you all for moving that science forward. Like I said, we definitely felt like we were going blind on that one.
Yeah. There is a question in the Q&A, and I would encourage folks to enter questions because we have just a minute left. The question was, is there a difference in your original study in outcomes? It looks like among the subgroup with metastatic cancer, if you have that answer at hand.
Yeah, there wasn't. We couldn't do a lot of subgroup analyses just because, our total sample size, but there really wasn't any difference by diagnosis.
Great. Other comments or questions? I was wondering a little bit about clinical trials registration and, clinicaltrials.org, and whether you have any particular experience with setting up deprescribing clinical trials and that requirement, if there was anything specific to that.
Yeah, I don't have anything specific to that. And aside from the fact that we always register our clinical trials, I guess maybe I would say this is one where, at least for us, having a good relationship with our clinical trials .gov liaison, or whatever they're called here at University of Colorado, has been very helpful, because intermittently you get those emails from clinicaltrials.gov that says that you're out of compliance with, at least I do, that you're out of compliance with something or another, and or we're due some reporting or something. And they've been our local liaison person here at University of Colorado has been so helpful in helping to navigate that, because I would say it's not entirely intuitive always.
We have just we're almost at the top of the hour. Mike, do you have a quick question that you wanted to ask?
Yeah, and there may be a long answer to this, so feel free to cut it short as need be. Thanks, Jean, for such an amazing presentation. One question I had is from thinking about the enrollment and refusals was what you learned about the experience of the deprescribing element and how patients perceive their willingness to join the trial. That is, deprescribing is taking something away, and was that perceived in a negative way that enrollment, and how did you handle that?
Yeah, I would say it was actually both from the primary treating physician as well as from the patient, and I would say the three buckets were, oh my God, you can't do this. Oh, thank goodness. And never thought about it. And I think from a patient perspective, thinking about it in terms of especially that language around what does it mean that maybe it's okay to stop this, and what does that mean in terms of my life expectancy. I think being really sensitive to that languaging and that messaging, like I mentioned, I think is really important, because it was everything, the patients who said, but my doctor said I have to take this for the rest of my life or I will die. Right. Because, think about what we do. I'm going to put on my primary care, what do we do while we're trying to motivate patients to stay adherent to their medications, right? This is the exact flip opposite of that. So being really thoughtful about some of that language. And then on the prescribing physician, honestly, it was everything from, oh my God, you can't do this, that's like malpractice, to, that's an interesting thought. Yeah, you're probably right. At this point, this patient may or may not need that medication. So I think understanding those perspectives, and I think it's going to be different medication by medication too. So it's probably where a lot of good footwork up front with key stakeholders, both prescribing providers and potential study participants, to understand where their headspace is around that particular class of medication is going to be important.
Wow, I'm still trying to take this all in, Jean. Thank you for sharing all of this. I watched you do this from afar, and I'm still in awe. So thank you for the transparency and integrity and the way that you've led this part of the deprescribing work forward. I'm continue to be inspired and impressed. Thank the rest of you, to all of you, for attending our webinar. A recording of this webinar is going to be posted on the U.S. Deprescribing Research Network website. And soon after we end here, we're going to send you an evaluation. So please send that and fill that out and give us our feedback. Our next webinar, we're still is to be determined, but keep an eye out on the network or in your emails for what that will be. Otherwise, thank you again, Jean, and thank you all for attending, and have a great rest of your day wherever you are.
All right. Thank you all.